Hi, everyone. Thanks for coming. Today, I will be talking about machine learning models and the machine learning revolution. And what my main message from this talk is that models are just a small part of the entire process of adopting machine learning and gaining value for the humanity. My name is Marcel Falkiewicz. I'm the leader of the data science team at Objectivity. I have a very diverse background, a very wide perspective. So I have a master's in psychology. I have a PhD in biology. I used to do brain imaging before I moved to business, doing data science commercially. So before we move on, I just wanted to mention that we have a small competition for you. You can win a small prize. It's 200 zlotys for any course on Udemy that you choose. And there's a link to two formulas. One is in Polish, the other one is English. Pick whichever one you want. So we are waiting for responses until the end of the day, and the winner will be the person with the highest score. If there's a tie, we're going to pick the one who did this first. So today I will make a longer introduction where I will tell you a little bit more about objectivity. I will tell you about machine learning revolution and different phases of machine learning. And then I will move on to a case study about fluorescence microscopy, and I will share a few conclusions from that case study and from that perspective. So moving on to objectivity. So this is a screen with just some of the clients that we had been working with over the last 30 years. So Primark is probably our oldest client. So in general, Objectivity is a software development company. We create bespoke software. Software development is the core of Objectivity, but we have also other services like data science, consulting, low code, and so forth. So we focus on long-term relationships with the clients. So a lot of these clients had been working with us for several years, and long-term relationships is essentially one of our core values. So moving on to the topic of today, I wanted to start off with talking about machine learning revolution. So in the last decade, we had seen some enormous advancements in terms of machine learning algorithms. So we had been able to solve problems that were either very difficult or impossible to solve beforehand. So just to give you a few examples, the AlphaGo that beat Lee Sedol, who was one of the best Go players in the world, it beat it 4-1 in a very public match. We have systems like AlphaStar, who plays StarCraft, a very complex game that people do professionally, and AlphaStar is currently able to beat most of the grandmasters that play that game. So the other advancements is Dota 2 is another game where reinforcement learning had been able to beat the professional players with a large margin. We have huge advancements in terms of object detection, which Krzysztof told about a little bit more in the previous talk. We have very large advancement in terms of machine translations, and then we have systems like AlphaFold, which predict protein 3D structure of proteins based on the DNA sequence. So we didn't find the artificial general intelligence. We are far away from that. So we didn't find the holy grail, but we found something like a holy hand grenade, something that is able to solve problems previously unsolvable. And that generates a lot of excitement. That generates a lot of hype, very high expectations about how this is going to impact our everyday life and the systems that we work with. So with machine learning, so it seems like the hype is fading away. a little bit, because there was a period a few years ago where there were very high expectations of the systems. People were engaged in very exciting projects, very ambitious projects, and these projects had failed. So if you don't know this chart, this is the chart created by Gartner, which puts technologies on different in different stages. So the first stage is the innovation trigger. Then you have the peak of inflated expectations. Everyone thinks this is going to solve everything. Then we have the trial of disillusionment. So this is the phase where people are disappointed and are more cautious about engaging with the technologies. Then we move to the slope of enlightenment, when people are realizing that there are some specific problems that can be solved and are using these tools more consciously with more realistic expectations. And then we have the plateau of productivity, where the technology is sort of stable and used as a standard thing. So if we look at the machine learning, it's so that this is from 2020, so this is 1 year old, and machine learning seems to be placed on this downward curve where people are a little bit disappointed that it cannot solve all of the problems that they expected to solve. So why do these projects fail? Why is it that the machine learning? But why is there disappointment about machine learning in some sense? Well, some projects, so I will not be able to go through all of the possible examples that you can give in here, but I picked one, which was the something called an AI super doctor. So this was a very ambitious project which was started back in 2014, I think, which was a mutual initiative between IBM, Watson, and PwC. So essentially that project, after 3 years and spending over $62 million on it, was scratched, was discontinued. The promise here would be to build an AI super doctor who would work with oncological patients. So the patients, so essentially the doctors are unable to go through all of the literature, all of the knowledge, all of the potential information that we can get from the internet in order to make decisions. So the premise was that, if we have a machine learning based system, it will be able to go through all of that information, synthesize it, and recommend the optimal treatment strategies for the patients. So the initial results were very promising. So they had very high concordance with the recommended therapies for one type of cancer. But then there were several moves, like the increase in funding, and then the scope was broadened, and it turned out that the system didn't work. It wasn't properly integrated. It turned out that when they tried to evaluate the system on a broader scale, it turned out that it didn't give so accurate results, and often the recommendations given by the system were just pure nonsense. They just didn't match any kind of knowledge by the doctors. So there were several other problems, but this was definitely an example of a very high hype, marketing associated with it, very high expectations, and then the confrontation with the reality that this system has a lot of shortcomings, and it would take much, much longer to develop a system that would work as expected. So this project could be called a failure. But I'll tell you about other projects that we did at Objectivity, which was not a failure, but something like an unrealized potential of machine learning. So the project was essentially a recommender system. We built that recommender. It gave very good results. So the subject matter experts who looked at the results told us that this is looking very good. This is very promising. We want to integrate that with our We have control over that system. It was another company who provided it, and we engaged with them, and they told us that there is just one way they could integrate that our recommender with their system, and that way was one of the most important features of that was that the recommendations weren't available to the users of the system immediately. They had to deviate by 2 clicks away from their regular process, what they had been doing over and over again with the original version of the system, in order to see the recommendations. And because of that, they just didn't want to do it. They had to build separate systems, separate monitoring, in order to encourage people to look at these recommendations, because it turned out that whatever they picked, like 80 or 85% of the time, it was also provided by our recommender. But they somehow did not want to go through these additional steps that were needed to get to the recommendations. So we have an excellent system that does what it's supposed to do. It's not a failure, as in this project. It was in concordance with the expectations. But because of the way it was provided to the users, it just wasn't adapted. So I see this as an unrealized potential. So the system had the potential to improve the business process by much, much more than it actually did. So now someone can ask a question like, what are the features of the AI systems, machine learning based systems, that actually do make impact, that actually change business process, change business, change humanity? How do you make impact with these systems? And PCG Gamma asked exactly that question. So they surveyed a lot of companies that were adapting or trying to adapt AI to figure out, like, what are the features of the systems? How are the AI systems that are successful integrated with the companies? And I highly recommend to read that paper. It's really interesting, even if you're purely technical and a data scientist, because it gives a very interesting perspective. But I will try to synthesize the most important points here. So I realize now that you might not see it. So essentially, there are 5 modes of operation of AI in some existing business processes. So one is AI decides and implements, AI decides, human implements, AI recommends and human decides, AI generates insights, human use it in a decision process, and then human generates and AI evaluates. So this is ranging from AI dominant and human dominant. So it turns out that the systems that generate the largest value are the recommender systems and the systems that generate some insights that humans use in their decision process. So essentially, this is analytics. The other ones, if AI decides and implements, it's significantly less successful. So this is another sort of look at this, but this is about how many modes are implemented within a specific company. So the companies that implement systems that cover all of these modes of deployment actually generate the highest benefit from an AI. And there's also another feature that is related to the system, is how the humans interact with the system. So essentially, so there could be an interaction where AI learns from human feedback. So it's the thing that you see on Facebook and everywhere. You click to remove an ad, and it asks you, why did you remove it? So that's the human feedback. This is the AI learning from you. Humans learn from AI. So this is another mode of operation where the ML AI system produces some recommendations, and humans try to rethink their way of acting in a given situation. And then there is a third method, which is the AI that autonomously learns from human behavior. So again, if there are systems which do all of these things, they're most likely to generate some value. So we have all of this information, and now what I wanted to show you is like how machine learning is delivered to people in some popular applications that were designed to be built to be for the masses, essentially Facebook, Instagram, YouTube, all of these applications that are that you use every day. They're essentially filled with machine learning to increase your user experience, to show you more relevant ads, and so forth. So these applications tend to be delivered. Here's an example of Whoop. This is a fitness application that essentially is showing you, so it's like a 24-hour heart rate monitor, and it's showing you some statistics related to how well you slept, how much are you rested, how much physical exercise you did, and so forth. It's one of my favorite applications, mobile applications. It's beautiful, it's very easy to use, it provides all of the information that is relevant at the given moment, despite the fact that it's filled with very advanced analytics behind the scenes. So that's what you get when you're a fitness user. This is something that people sell. And now I'll show you two more pictures from a software that I used to work with back when I was doing brain imaging. So this is an interface to one software to another software. I won't call the names. If someone recognizes it, please don't tell anything. But this is how complicated and how challenging using of such software is. So I really spent a few weeks learning how to interact with the software because I needed to do something. And the idea, and I checked also on their website, they have an easy to use version of this, which is a shell script that you essentially run somewhere. So this is packed with very advanced analytics, very complicated things, but it's also its interface, the way it's delivered to the people is so complicated that PhD students who do brain research, they have to spend weeks, months learning that software, learning to interact with that software. So it's very hard for them. So it's very hard for them to interact with it, to gain some insights, and they often make mistakes because the interface, the entire thought process behind using this is based on people who focus on methodology, and they believe that it should look like that. You should have all of these options available to you.
You should understand everything by heart. And
yes. Yes. So there's an anecdote. We had a recruitment. There was a person being recruited, and he said we talked with him about the interfaces for the software, and he said that he had one interview, and they asked him, what do you think about the UIs? And he said, I want to build applications that have a very nice UI. And they said, this is scientific. We are producing scientific software. It has to work. It doesn't have to look. Thank you very much. And that was the end of his challenge to get recruited by that company. So this is really the approach. So I'll show you. And can we marry these worlds somehow? Can we produce software that is easy to use but does quite advanced things by the subject matter experts? So can we help them? Can we somehow get rid of all of that and build something that will clear up the way to be more productive in scientific applications? So and I'm moving on to the case study that I wanted to show you. So essentially, I want to give you a context. So we work with a drug research company that does early stage drug research. So they're essentially looking for candidates for medicine. And I'll give you a 1-minute crash course on how do you find drugs. So please bear with me. So essentially what you're doing, so one of the ways of finding drugs is you have some chemists who are synthesizing some compounds that they believe would work for some kind of a disease. You take these compounds, and in the very first stage, you put them on something called the microplates. So these are just like small plates, and here there are some small holes in those plates. These are called wells. So you grow artificial cells in those wells, and then you treat those cells with the compounds that the chemists had created. And you can, so if you, here you have the plate layout, and in the columns you have different drugs or combinations of drugs, and in the rows you have different concentrations. So here you have the highest concentration, lower, 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 and here is the lowest. So that's essentially how people, how they look at the cells, and they're looking at the cells' reactions to the compounds, asking different scientific questions. So they select the most potent, the most efficient drug, and then they carry on with testing this on animals. Of course, if there is information that it works. So in reality, so if we unpack this bit where they actually select something, they're putting these microplates in a microscope. This is a microscope, believe it or not, which is doing something called high throughput imaging. So it's essentially doing a photograph of each of these wells in several places. And also these cells are treated with fluorescent dyes, which highlight different compartments of the cells. What you get in the end is these beautiful pictures. And these pictures, well, they depict cells and different compartments of the cells. So here you see in blue, you see the nuclei, and here you see other compartments of this. This seems to be tubulin, but I might be lying in here. We have pictures like that, and there's hundreds of these pictures, and there's a lot of information in the data. So what the scientists are interested in is finding phenotypes. So in this example, we would say that one phenotype is like one way the drug worked on the cell. So for instance, we could have one phenotype would be the cells that have this brighter skeleton around it, and the other phenotype would be the one that is without this bright part, just some faint shadow around the nucleus. So this would be one phenotype. This would be the other phenotype. So the entire trick is that, you know, if you have hundreds of thousands of these cells, you can't go through all of that data when you're a scientist. In the end, they want to see, depending on the concentration, how were these cells affected? How many percent of the cells, for instance, express this phenotype, depending on the condition, on the concentration. So we will look at this row. So they produce something called the dose response curves. So they used to do it in a way that employed classical computer vision, which was essentially a set of filters that they had to build in order to isolate the interesting phenotypes. So for instance, they could count all of the nuclei in the image. That would give you the total number of cells. And then they built filters that left only some parts of this phenotype and leaving none of that phenotype on the images, and then counted these. So you would have the proportion of cells. So it's essentially counting. You had to count these and compare it to the total number of the cells. So because they use the classical computer vision tools, and for every single data set, they had to build a separate pipeline. They had to do it all over again. It required expert knowledge. There was only one person in the company that did this really well, and she left, by the way, during the pandemic. So there was a lot of difficulties associated with this. Yeah, yeah. So it was really a huge problem that these pipelines, sometimes they couldn't just figure out what filters they should use. Sometimes it took them a weekend or 2 weekends because, you know, they had to leave their laptop running these analysis. Sometimes it crashed in the meantime. So it was a huge pain to analyze that data. So we came up with a solution based to replace that system with some more modern machine learning. So essentially, the architecture of that system, it's simplified in here, but I'll just give you a preview. So essentially, when we had these images, we had to find the nuclei, so these objects. So this is pretty easy, but well, apparently easy. It's not that easy if you think about this. We had to find the nuclei of the images. So we used an object detection network for this particular task to segment the nuclei. Then you had to cut out the entire cells. So if you have a bounding box around the cells, you have to cut it. You have to cut out the entire cell because the nucleus is in the center of the cell. And there goes the whole analytics pipeline, which involves a autoencoder, or we have the other option to use a self-supervised learning model, which essentially turns images into a small number of features or numbers. So we get a small vector for each image that describes it numerically. And then we are doing dimensionality reduction in order to get a two-dimensional representation of those cells. And then the scientist can look at those points on a 2D plane and find groups that hopefully represent different biologically meaning phenotypes. So we have a object detection here. We have an autoencoder, so some form of self-supervised or unsupervised learning in here. We have dimensionality reduction. What is not on this diagram is active learning. So we have also a supervised mode where the scientist actually looks at some of the cells and tries to label them. And then in the end, it's all about just labeling the points and getting some kind of a report in there. So if we look at that case when we started to talking about UI, about UX and so forth, they didn't know what we are talking about. So they never had in mind that a software like this can be produced and can be easy to use. But we did it, and I'm really unhappy that this looks like that, because it's high resolution in reality. So I'll try to describe what we have in here. So here we have, so we designed it. So these are the screens from a development version of the application. So this is the screen where you have just the list of the data sets or assays, if you like, that you can choose. You can pick one, and you can see its status. Once you enter some session, you see some data set, you see a list of analytical sessions that are associated with that. So this is just a list. You can pick whichever you want. You can name them. You see the owner, when it was created, modified, and the status, like, what's going on in the analysis after that. This is the screen that, I'm really sorry that it looks like that. So, on this screen, you can essentially pick the bounding box size. So you can see, so this is already after the object detection. So this runs in the cloud, in the background. So you don't have to do anything. You just click the button. It spawns a computational node in the cloud and does the nuclei detection. On this screen, you also already see the results. You see the bounding boxes, and you can enlarge them to include the entire cell bodies based on your actual images. And then you move on to the model setup. And as you can see, you have the option to reuse models, to create new models, and so forth. But there's just like 2 or 3, 4 options that you need to pick from just to start using that system. So once you click that, it essentially creates a numerical representation of the cells that were extracted previously using either self-supervised model or a variational autoencoder. You can pick active learning mode, or you can just jump straight to the scatter plot. So if we look at the scatter plot, we have the representation of the points. You can interact with this plot. You can essentially mark them. You can assign labels. You can regenerate the plot, because behind the scenes we have an algorithm called UMAP, which is a quite advanced dimensionality reduction algorithm, which essentially tries to find the best topology that, the best representation that recovers the high dimensional topology of the data points, and it tries to separate the groups. Here we have 3 phenotypes, and we can see that this is quite nicely separating them without any supervision, without any input. So the scientist can sit down here, he can look at specific points, zoom in, zoom out, he can look at the specific images, look at the metadata, and he also has the preview of how the plate will look like, how many. So if he assigns the classes to specific cells, he will see the entire plate and see how many cells are in the particular wells on that microplate. And by doing this, so by doing this, we had built in quite a complicated analytical pipeline built on Azure with on Azure machine learning with several machine learning models, with these models being retrained almost every time someone does this analysis. But it doesn't require the scientists to know what they should know what machine learning is, but they don't have to have another PhD in machine learning in order to be able to use that system. So this is the development. So these screens are from the development version. It's still a work in progress, and we are moving towards the MVP phase. So what are the conclusions for this? So I believe that the value of machine learning will be fully realized if we can give it to domain experts. Not necessarily technical people, not necessarily people who know what machine learning is, but if we give it to them in a way that is approachable for them and will allow them to integrate these tools with their everyday life. If we move out of this hype phase, this will be very good for the discipline, because then we will be able to focus on where can we actually make value, on building some products that are actually working and are not just delivering high hopes and high expectations that are likely to hurt the entire field. And apart from further model development, we should focus on delivery, because if we deliver the models to the users, then we discover things that we would never imagine. So then it turns out that people are entering some input that you never expected. There are data points that are completely unexpected. There are weird behaviors that no one understands. And this allows us to iterate through the model development process further. The more seamless the integration is, so I already mentioned this. So if we manage to integrate this with experts' work, deliver it precisely where it is needed, then the impact of those systems will be higher. We will be able to treat disease better. We will be able to find better drugs faster and do other amazing stuff that machine learning allows us to do. And finally, the learning process should be mutual. So we should provide feedback. So when we're building machine learning models, machine learning systems, we should be collecting feedback, user feedback, because this is invaluable to making any improvements. But also, we will know that the machine learning system is valuable when the humans are going to learn from it. So for instance, I don't know if you followed the AlphaGo story, but Lee Sedol, who was retired at the time when he played with AlphaGo, he said that these matches with the machine actually respawned like his love for the game, and he gained some new insights into the game because of those matches, because of how he played. This is a very narrow world, so this is not something that changes humanity forever. This is just like one game. But this is the kind of impact. You know that the system works, that it produces something amazing, and it's very well designed if it does this kind of impact. And that's pretty much it, what I wanted to say. So thank you very much for listening to my talk. And if you have any questions, I'll be happy to answer.